the International Congress of Parasitology, and it's being held in Montreal, Canada, from August 16 to 21. And ICOPA is organized by the World Federation of Parasitologists, and it's organized every 4 years. And the theme for this year is Parasites in a Changing World. I think quite a number of us have already registered for the Congress, and the registration has been extended up until the 3rd of May. So we still have some more days to register for the Congress. And as part of this year's Congress, we decided that we would have periodic webinars, and what we're doing today is one of them. The theme so I spoke about this already, that the World Federation of Parasitologists is organizing the ICOPA. And as part of the Congress, we decided that we would hold periodic webinars, and this webinar is one of them. And the theme for this webinar is Malaria Elimination Roadmap, the what, how, and when. And this is as part of our commemoration of the World Malaria Day, which is held on the 25th of April every year. So today we'll have 3 speakers talking for about 15 minutes each. The first talk will be a talk on vector biology, and the title is Leveraging Vector Biology to Accelerate.
Laptop. Where?
My screen went off. To accelerate the elimination timeline. After that, we'll have a talk titled Understanding Immunity Exposure and Infection Persistence for Malaria Elimination. And then the last talk will be Winning the Battle, Losing the War, Parasite Adaptation Under Intervention Pressure. So today we have 3 speakers, and then we want to have quite a lot of answers. So the format would be all the 3 speakers will speak, and then the last 15 minutes would be dedicated to questions and answers. So we'll be a little strict with time, just so that we all benefit by having our questions answered. So without talking too much, I would welcome Dr. Joelna Okoli to give us the talk on leveraging vector biology to accelerate the elimination timeline. Thank you very much.
Thank you so much, Linda. I will, hello everybody. I will quickly share my screen. I'm used to using 2 screens, so I hope I don't mess up. Just a second. Share. Okay. Oh, okay. Now this is becoming a problem as always. I did try this. I'm so sorry. Just a minute. Okay, there we go. Can you please see my screen now?
Yes.
Thank you
Not in
very much.
presentation mode, but perfect.
Oh,
perfect.
It's in presentation mode now, isn't it?
It is perfect.
Thank
Yes.
you. Thank you. So thank you, and good afternoon, good day to everyone. My name is Joelna, and today I'd like to talk a bit about vector biology and the role it's playing in malaria, and the road to elimination, and what vector biologists as we are, and the role we can play in that roadmap. We know malaria right from when we were kids and when we're going to school, that malaria is a vector-borne disease. And that only means that the disease can only be transferred from one person to another through the bite of an Anopheles, female Anopheles mosquito. However, we don't have to quickly lose track of the fact that there are other drivers of malaria transmission. Yes, the malaria vector is probably the most important one here, but we live in an environment where the vector interacts with the environment. We interact as humans with the environment. And so it becomes a One Health issue. It becomes a connected and probably a bit more complicated issue, as we used to think of it as it's just the mosquito and the parasite. Since the 19th century, when we first discovered that mosquito was the main vector of malaria, we have made up our minds to try and understand the mosquito and how we can target it for malaria elimination. Our idea is that once we get rid of the vector, then everything should be fine. And so how we've been doing this over the years, over so many years, has been to use chemicals. We have tried the insecticide bed nets to prevent mosquito contact with humans. We have used the indoor residual spraying to try and kill them on target. And then even larviciding to try and remove them as larvae or imagos or immature forms in their breeding site. And what this has caused is simply an evolutionary arms race, basically. We as the humans are bombarding the mosquito with all the arsenals that we have. And what the mosquito, smart as it is, and we didn't really know this from the beginning, was that the mosquito is smart enough to come back and also attack us in some way, because we attack it. It goes through some evolutionary process because it's also an organism and it's also trying to survive. And so far, what it has caused is just that we're just going around the issue. We hit it with something and it comes back fighting, and even stronger than when we started. What this has caused is basically a gap in the milestone that we hope to achieve. Now, what you're seeing here is a graph I took from the current WHO malaria report. The blue line you see is our current status. The green line is where we had hoped to be by the year 2030. And what this is basically showing is that the milestone we had aimed to reach we are pretty much far from it. The gap is actually it's widening. And as at 2025 or 2024, this is where we are, and it's probably going to open much wider by 2030 if we don't do something. Like I said, it's a complex issue now. It's not just about the mosquito. When it comes to the mosquito, basically our major factor, our major problem is insecticide resistance, because it's probably the only weapon we had was to treat it with chemicals, try to get rid of it with chemicals. So apart from using these chemicals, we were almost like lost as to what else we could do until quite recently when new tools started coming up. And that is to say that our toolbox or our toolkit has shrunk, and we need to now go beyond chemicals and try to find new ways of getting rid of this disease. Oh, sorry. And then there is also the emerging threat of what we call the invasive vector species. There are about over 400 species of Anopheles mosquitoes, and they are distributed all over the world in different prevalences or different proportions. And so there are some mosquitoes that are quite restricted to certain areas of the world or the globe, and basically they are playing their own role in transmitting malaria in other places. However, because of global trade and the fact that now things are quite open, we move goods and people across sectors, across countries. This is now becoming a challenge because we are gradually seeing new species of mosquitoes that are in the past restricted to some areas moving across borders into new niches. And this is becoming an increasing threat because very soon we will be in a situation, or if we are not already in that, a situation where the current arsenals that we have, the current weapons that we have localized within a geographic region may not be working anymore because new vector species are coming into the area. And then the almighty one, which is looming and all around us, is climate change and environmental change. We are now have to target this. We are in contention with this one, and it's nature. We probably have caused it, and we now have to deal with it. And the increasing rainfall, drought, and storms are creating new niches for mosquitoes in places that they weren't already existing. And so these are some of the three things that I can identify broadly that are widening our milestone gap or our target. In Ghana, putting things into a bit more perspective, as I've said, our main chemical of use had been the pyrethroids and to some extent the organophosphates. And this is a study that the Vector Research Group of the Noguchi Memorial Institute for Medical Research had done in collaboration with the National Malaria Elimination Program and Ghana Health Service over the past 10 years, over 10 years. And what they had done was picked 30 sentinel sites across the country, as you see here, and have monitored insecticide resistance status from, I think, around 2015 till date. And what you see here is the red dots are basically confirmed resistance of our mosquito species to pyrethroids. So immediately you see that our major chemical of use, the mosquitoes are really resistant to them all over the country. To some extent, organophosphates gives us some breathing space because then we probably may have some chemical or some insecticide to use because there are other there are some places in the north, for example, that have still mosquitoes that are susceptible to organophosphate. But this was about 5 years ago. Currently, the current reports that I received or I have come across was reporting data on 2023. The pyrethroids status hasn't changed at all. We still are seeing across the 30 sentinel sites and across 4 different pyrethroids that were tested, we are seeing low mortality. The mosquitoes are not really dying. What we expect to see is anything above 90, really. But you see that on average, our mosquitoes, 10% of our mosquitoes, only about 10% are being killed, and which is still a concern. Then we have some glimmer of hope because we have a synergist called the PBO, and this is a chemical that basically interacts with the metabolic enzymes that would ideally give resistance to the mosquito against pyrethroids. And we're seeing that with the addition of the PBO, it depends on the southern sector of the country. We are still not seeing that drive the mortality that we want, but there are some places in the northern sector that we are seeing that with the addition of the synergist, the mosquitoes are actually dying out of the effect of the pyrethroids. There are new formulations that are targeting new sites, new central neurological sites of the mosquito. However, although you see all the bars going above 90 as we expected, I'm sure you also see there are some sites that have already been reporting resistance. I mean, it's like our arms race, like I said before, is starting yet again. I did mention that one of the things we need to struggle with, most people are afraid of and mentioning, it's invasive species, new species coming into our sector, our location, other than the ones we already knew. And the key one here is the Anopheles stephensii. Anopheles stephensii, this plot I'm showing here, or this picture I'm showing here, they are all Anopheles, between Anopheles gambii and Anopheles stephensii. They are all Anopheles. They are morphologically distinguishable, but this has to be done by an expert an entomologist would have to sit and look at it, their wing patterns, as you can see here. So I'm showing with the red arrows how different sectors or parts of the wings are between the two species, and even the so you see patches of white here, but you see dotted patches for the Stevens eye. So they can't be morphologically distinguished, and they can't also be distinguished molecularly by PCR. And actually, that is what happened as at 2023, where a group here at the University of Ghana, Prof. Afrani's group, the medical school, also detected Anopheles stephensi here in Ghana. And that quickly added Ghana to the very many countries across the African sub-region that have started reporting Anopheles stephensi. Stephensi was initially restricted to the Southeast Asians, but now we're seeing it come into West Africa and Africa, to be precise. We see it in Ethiopia, in Kenya, in Sudan. Nigeria has also reported it. So what did we do as a vector research group of Noguchi? We have a student, a brilliant master student, Ramat, who's currently doing a PhD, who nicely took climate data, and modeled against the climate across Ghana to see the environmental suitability of Anopheles stephensi. Now, you see here in red color where it is very suitable for a stephensi to establish itself against places that are much greener. So the bar is from green, which is red, zero, up to red, where it shows that predictively these sites are places that could potentially or are very ripe for Anopheles stephensi establishing itself. And this only goes to show that there are still a lot we need to learn about this new species. The ecology of it, we still don't know much about in terms of Ghana. We don't know much about its feeding patterns. We need to know the vector biology so that we will be able to know where to find it and how to target it. We don't even know yet whether its insecticide resistance is similar to what we already know for Anopheles gambiae across Ghana, and whether it is susceptible to our local parasites. Like I said, it came from Southeast Asia. They have vivax, they have mostly vivax, and Plasmodium vivax, sorry. And we don't yet know how susceptible these Anopheles stephensi, if they should come to Ghana, will be to our local parasites. And these are still all areas that we still need to investigate. In terms of climate and Ghanaian landscape, this is a paper I liked seeing. I saw it and I was excited to see that our climate scientists are also doing something in terms of checking how our climate and environmental landscape is changing. Going from left, on my left to right, we're seeing what the situation, how things look like down in Accra here. So you see airport, you see Tema. How things have changed over time, how our land cover is changing right from 1991 to 2017. You see how red things are becoming. That's because we are urbanization, we are building more, we are cutting down more trees, we are clearing our land for various other things. And that is also actually increasing our land surface temperature. So we are very much at risk of the climate heating up. Things are going to get heated up. It's probably even worse now in almost 10 years after this paper, or this data was shown. So I would say Ghana is at a point where we need to be very critical about how we look at things in terms of malaria elimination and for all other diseases, to be fair. How must we learn? We have to be smart. And the whole point is to go ahead of the arms race. We need to go ahead of the vector. And these are a few things that we can do. I'm sure there could be more. And that we need to sequence. We've learned from COVID that if we sequence as we go along, if we sequence in real time, then we go ahead of the parasite. Here, I'm talking about going ahead of the vector. So we need to track a bit more. We should move away from it is there, it is not there, present, absence. to tracking it. We are bringing in new formulations. Have we thought of tracking it? How the new formulations we are applying on the field are changing the mosquito? How are they adapting to it? Are some of the things we need to start looking at. We also need to look at the environmental conditions. It is not just the vector biting the mosquito, human being and bringing disease. It is the environment. Our land cover is changing. We are urbanization, urban agriculture, and rainfall patterns are changing. So we need to also bring in our climate scientists. We don't just need to just do our vector. The vector is very much connected to the climate. It's only when it rains little or much is when our breeding sites will change. The temperature would accelerate mosquito development. So those are some of the things we need to adapt. And then we have tracking also our vector susceptibility to emerging parasites. I'm sure Prof. Sagansa will talk much more about the parasite emergence and surveillance, but we also need to do this together. We cannot do things in silo. The vector surveillance and tracking would have to go with the parasite surveillance and how things are changing across all the factors. Like I said, we have to be climate smart. Things that it has to be climate model. Our models can't leave climate behind. So I will end here with some work that the Vector Group has also been doing. We need to expand our toolbox. And one thing we have been looking at now is microbes. I'm sure this may sound contradictory to AMR, but mosquitoes also have microbes. And the microbes we have found over time do a lot of interruption of the parasites within the vector. So we are thinking of transmission blocking strategies using microbes. And the group at Vector, the Vector Research Group at Noguchi has done something. We've started to look at the microbes. And that is, putting in microbes and seeing. So this is a plot that is showing oocyst number of parasites reducing in the mosquito, and when we have added in a bacterial product. So I will continue by saying that we need to move from lab to policy. We cannot just work as vector biologists. We need to train a bit more entomologists. People need to be trained on the new trends of disease identification, surveillance, and data analysis. And all our evidence would need to go to policy. We don't need to sit on our data. We need to communicate very well with the National Malaria Elimination Program, as we've already been doing, and all other stakeholders that are involved. So in summary, I'd say we need to monitor, we need to be innovative, and we need to adapt to the growing trends of climate change. So I will end here by acknowledging all our partners. I hope I haven't missed out anything, but if I have, we thank you if you're a partner, a sponsor, a funder who has supported our work as a Vector Research Group. And thank you. We are committed to eliminating malaria as vector biologists. Thank you very much.
All right. So thank you very much, Joelna, for your talk. I think we have a lot of questions, but we really need to save them for the very end, else we may get into trouble. So thank you so much, Joelna, for the talk on leveraging vector biology to accelerate the elimination timeline. So there's a lot that needs to be done with respect to the vector. Now we have Anna telling us about how we should understand immunity and exposure and infection persistence to enhance malaria elimination. Anna, please do note you have 15 minutes. Thank you very much.
Mute. Thank you for the invitation. It's lovely to be here with you. I'm from Stockholm at Karolinska Institutet. And I think we've been already reminded that it's really the children in Africa that are most affected by malaria. But I think now it's not only the youngest, it's also other age groups we need to consider when we manage and think about malaria and elimination. So this map and these numbers are really telling us that we're far from eliminating malaria. But there are successes in some areas, and locally, malaria is about to be eliminated, but in many areas also now increasing. And we don't know with the current situation, funding and so, how it will look in the coming reports. But we had some groundbreaking interventions coming in with the 2 now vaccines being rolled out in 2 small children in sub-Saharan Africa. So these 2 vaccines are partly efficacious, and we're looking, but still we believe that they will affect the morbidity and mortality in those children, and maybe on a larger scale, but we're still to see more of the impact as they are now being rolled out. And to make better vaccines, we need to understand immunity. And immunity is then also very much depending on transmission intensity. So in areas with high transmission, immunity is acquired, or the clinical presentation will look very different. But what's clear is that although immunity is developed, it's also difficult to clear parasites completely, and especially asymptomatic infections are therefore highly prevalent in many areas. So this is another map then of the world and the parasite prevalence in parasite rates in children 2 to 10 years old. So you see more than 50% of children are actually carrying malaria parasites in their blood without necessarily having symptoms, but I think more there's anemia and other health consequences that need to be considered. So what I share with you today is some of the work we've done both in Tanzania and then also what we can actually do in Sweden to contribute to the elimination of malaria. So we've been following a village in the Rufiji River Delta, coast of Tanzania, since 1985, where the population is participating in cross-sectional surveys. And we noted already 2010 a decline, which was also reported across in other sites of Africa. We have then continued to follow this village and see that it's actually the Plasmodium falciparum that declines the most in these surveys. And I should say that we have distributed both the research group team, but also a couple of governmental distribution of insecticide-treated nets. So these are unpublished data, where you can see then from 1986 until 2024 how dramatically malaria has declined in this village. So from being a whole endemic area to now being almost pre-elimination. But we are worried about what's going to happen, because we don't know the clinical consequences yet, and the question is what has changed. So there's a lot of research to be done to understand this decline, but we believe bed nets are important. When we then go into immunity, which is our focus for research, and we looked at antibody responses in the high and low transmission period, and you can clearly see that just looking at the whole antibody responses to whole falciparum chicons, they are across ages much higher in the high transmission period than in the low transmission period. And also antibody function. So in this case, antibody-dependent respiratory burst is much lower in the low transmission period than in the high transmission period. And what you see on the right, the left-hand side is antibody responses to different merozoite surface antigens, which we think are important to be immune to malaria. That it's not, and that also comes back to designing a malaria vaccine. So maybe not, or preferably including several antigens and making a multi-component vaccine. So these blood stage antigens are likely to be highly important. So what can we do to understand what's happening with immunity to malaria in Sweden? So this is a normal winter here with snow, and we only have about 200 malaria cases diagnosed, clinical cases diagnosed in Sweden per year. But what we realized is that we can actually study the duration of immune responses to malaria without re-exposure, because we know that people will not be infected again. So we both study clinical aspects, but also immunology. And just to share with you some previous data on clinical malaria, where we saw that people on the green bar with origin from sub-Saharan Africa are less severely ill in falciparum malaria than Swedish travelers. But after 15 years residing in Sweden, they end up in the intensive care just as often as a non-immune Swede. So immunity to severe malaria is not lifelong. And then we built a cohort of people that we invite, the patients that we invited at the time they were admitted with falciparum malaria, and they then contributed with blood samples and done so several times over 1 year. And here we have the possibility to compare a Swedish or European traveler who had malaria for the absolute first time with patients that have been born and lived many years in malaria endemic areas, mainly sub-Saharan Africa, and then living in Sweden and going back. So we can look at the importance of previous exposure memory. So we do a lot of immunology in this cohort, and then can with different modalities looking at both antibody and cellular responses, and more recently proteomics. That I won't go into all that today, but just share with you that it's clear that the African-born patients, the gray bar, have higher and broader antibody responses than the first-time infected Swedish traveler. Also looking with a broader array with 111 merozoite surface antigens, we see this pattern, and so the breadth of the antibody response and the magnitude is higher in previously exposed. Using this type of setup, it's almost like an experimental setup, since we know that people are not re-exposed. We can also model data, and in this case, we identified serological markers for recent exposure that then could be used in monitoring malaria transmission in elimination settings. We have looked at memory B cells and are doing that with different methods. This was actually one of the first malaria immunology studies we did in travelers, where we saw that even after a single infection, a Swedish traveler will have circulating memory B cells that recognize malaria antigens. So it's not that the memory is not built, but it's not good enough, or the antibody titers are not good enough to protect people. And we then also did more sophisticated method to look at more those merozoite surface antigens at the same time, and comparing the groups. And developing these methods, we can then go back, in this case, in a study in Kilifi, Kenya, to look at the memory B cell responses with this fluorospot method, and shown that the breadth of memory B cell responses is correlated or associated with less clinical malaria in children. So that was just briefly the kind of things we can do in a setting where we don't have transmission. Another thing is to look at duration of asymptomatic infections without re-exposure. And this was actually a surprise to us, although we worked on asymptomatic infections for many years, and different aspects of of that. But so what we did was we have invited people that have arrived to Sweden from sub-Saharan Africa, mainly refugees, but also other migrants, and invited them to be screened for parasite asymptomatic infections. And overall, including all countries in sub-Saharan Africa, all ages, we had a PCR positivity of 9%. And when we look specifically at the different groups, we actually found that those that came from the highest endemic countries have children, up to 36% of children and 23% of adults were positive for malaria parasites by PCR. This is also unpublished data, but we've looked at newly arrived refugees coming to Sweden to certain regions in Sweden, and in there we can actually see up to 45% parasite prevalence in this migrant group. So we clearly have asymptomatic infections that persist after arrival also to a non-endemic area. And now the question we are working on is both to understand the clinical impact of these infections, but also the duration. And in the first publication, which included about 800 migrants, we looked in the subset coming from the high infected group and found that people had infections after 1 year. But now we are setting up a study where always ongoing, where we actually include and invite people who have lived here for more than a year, and have now several people, parasite positive individuals that have lived here more than 2 years. And the longest so far is an asymptomatic infection, 5 years after infection. And although it is reported as case reports that many years can have gone since exposure, this has not been systematically studied. So we believe this is a very interesting setup to study this. And this is very preliminary, but we'll be happy to share more as we go along with this. Within this asymptomatic infections, we also study the antibody response and look at the kinetics and the longevity before and after treatment. Because we should say all the if we find parasites, everyone is treated as soon as possible. But just here, looking at the antibody response against Schizont extract of Plasmodium falciparum, it's also clear that PCR positive individuals have higher antibody titers than PCR negative. So in conclusion, immunity wanes without repeated exposure. There is likely a risk of rebound in transmission in an area like Tanzania, and therefore also risk for clinical and severe malaria. So it's very important how we monitor the malaria situation when it declines. And then finally, asymptomatic infections may remain for years, and that may is then a challenge for elimination efforts. And to conclude also, I would like to say that we really need those our global efforts to eliminate malaria. And if we ask the questions, we can try to answer them in different ways. And these were some of the examples. So, yes, thank you. Just finally, of course, the acknowledgement slide, and I want to acknowledge especially Doreen Mutemi at the Muhimbili University College of Health and Health Science in Muhas in Dar es Salaam, and Victor Iman at Karolinska. And Doreen just defended her thesis on the work I showed from Tanzania, so we're very happy for that. And also would like to acknowledge Dr. Inga Drew to set up the study in Nyamisati in Tanzania. Thank you.
Thank you very much, Anna. This was a very nice talk on immunity. So thank you so much for that. We've learned a lot about how immunity is developed and the role asymptomatics play in the development of immunity. We'll now give the stage to Anita to tell us about winning the battle or losing the war, parasite adaptation and intervention pressure.
Thank you very much, Linda. Good day to all of us. So my talk is on winning the battle, losing the war, the parasite adaptation, and intervention pressure. My name is Anita Gansa from Noguchi. Yeah, so we've had a lot of background, but just to add that we've had malaria cases and deaths averted over the past 20 years or so. We've seen over 2 million malaria cases and 12.7 million deaths averted. And this is all driven by widespread deployment of the vector control interventions, ACTs, and testing with RDTs. Now, Ghana has also chalked a lot of success. We have moved from about 27% prevalence in children under 5 to 8.5% between 2011 and 2012 as a result of these interventions. But we know also that these successes have stalled, and in fact, the global case incidence increased by 8.5% since 2015. And there are a number of issues that have resulted in this, including lack of funding or donor fatigue. COVID has played its role, but also we have the biological factors. Joanna has looked at vector control and insecticide resistance. Anna has looked at immunity. So I'm going to focus on how interventions are shaping the parasite population as an evolutionary force. So every intervention changes the rules of survival for the parasite as they impose selection pressures that the parasite would continually adapt to. And this eventually will reshape the parasite's architecture. And so we will look at how the parasite is shaped within the vector or with vector control interventions, and also with clinical interventions like the drugs and diagnostics. So in general, how would the parasite develop in the mosquito? Without any intervention, we will see the normal development of the parasite. As the mosquito picks it, it will go through its developmental stages, and the relationship looks more like a commensal one, that the parasite will have the opportunity to thrive within the mosquito, and the mosquito will remain neutral. But when there are interventions such as the insecticide exposure, either through indoor residual spraying or the long-lasting bed nets, it shrinks the mosquito population.
And how there? She's working on a lot of things. So unless she's done, then she calls me to.
And the mosquitoes will adapt in a way that helps them to shorten their lifespan. So they tend to produce gametocytes more quickly. And the whole developmental process is affected because there is an immune pressure exerted by these mosquitoes that are surviving insecticides. And this immune pressure is exerted on the parasite as it tries to grow. And so it causes a severe bottleneck within the parasite population. And remember that it is within the mosquito stage that the parasite undergoes sexual recombination during zygote formation. And this is how the parasites introduce extensive genetic diversity for its survival. So yes, the process will go on, but it is all reduced, even though there's faster development of these parasites as an adaptation by the mosquito to sort of go through its life cycle before it is taken away. There's also the immune evasion, which reduces the parasite population. And so the resulting parasites from this type of situation will be theta and selected, and these will optimize transmission. Now, in the case of the drug, in the absence of a drug pressure, again, an infected mosquito bite would go through the parasite's life cycle within the host up to gametocyte formation. And the drug, like the artemisinin combination therapy, will rapidly kill the young rings, and the partner drugs will also clear the remaining parasites. And so all parasites will be cleared and transmission is minimized. So high parasite clearance, low selection pressure when the drug pressure is very low. But in a situation where there is repeated use of drugs or partial killing of parasites as a result of suboptimal drug doses, malabsorption of drugs, or drug-drug interactions that still leave some drugs in circulation, or counterfeit drugs, drug pressure will develop. And the parasites will respond to these drug pressures either through a genetic resistance or a mutation that is not reversible immediately, resulting in parasites that are resistant to drugs, or they will undergo maybe copy number increases or variations in response to such selection pressure. There's also the activation of processes like stress response, where the parasite may adapt a dormant form, especially in the ring stage of the parasite, to avoid or delay the clearance of the parasite. As we know, artemisinin works best in the early trophs or the trophozoite stage. So if you delay ring stage development into trophs, you delay clearance of the parasite. You can also have expression of gene changes in expression in response to these stresses, or epigenetic changes as well. So it's a number of processes that the parasite will induce. Now, the outcome of all these will be delayed parasite clearance or higher recrudescence risk, and greater transmission and spread of resistant lineages in the presence of these drug pressures. And we know that the main driver of artemisinin resistance is the KELCH13 gene and its associated markers. Now, the K13 mutation is driving artemisinin partial resistance, and the phenotype is delayed clearance. And we know that it emerged in the greater Mekong region and has spread through Southeast Asia. And now we are seeing independent occurrences of these markers from the Horn of Africa, and it's spreading through East Africa, and it's coming. But there are also non-canonical agents or other genes that typically will not be activated, but they are not related to the main KELCH13 or the main driver of artemisinin resistance per se, but they are also activated in response to the drug pressure. And the situation in Ghana is that as we monitor the emergence and spread of artemisinin resistance, we are finding just isolates of these markers of artemisinin resistance. They've not been established, and we have very efficacious artemisinin-based combination therapy, over 95% efficacy for all our drugs. So we do not have artemisinin partial resistance in the country. Now, in terms of the markers of diagnostic resistance, the PFHRP2 or 3 gene, or the protein, is used to develop the rapid diagnostic test for detection of malaria parasites. And the parasite, again, has adapted by evading diagnostic, by deleting these genes either partially or fully. And as a result, you will get a false negative test result if there is a deletion. So this is the false negative results. And you could also get a false positive results due to residual PFHRP2 or 3 proteins in the bloodstream that tend to remain in circulation for a few weeks after treatment. Another scenario of getting a bad diagnostic result is when the parasite is Plasmodium falciparum, but with a deleted HRP2 in a mixed infection with a non-falciparum parasites. So because the HRP2 is Plasmodium falciparum specific, these non-falciparum parasites will not be detected and results in a false negative. And WHO has set 5% as a threshold for changing the diagnostic test kit. And at Noguchi, led by Professor Linda Amwa, we are monitoring the spread of HRP2/3 gene deletion. And I think Linda will tell us more, but I know that we are nowhere near this 5%. Right. Now, for the vaccine, we know that antigenic diversity will have a huge impact on the parasite population or the vaccine efficacy. Allele-specific response to vaccine will also have an impact. Immune escape are some of the reasons why the vaccine may not work. We at Noguchi have established that the CSP epitope that is being used to formulate the RTSS or R21 vaccine is extensively diverse across the Ghanaian population. So these are 2 populations, Cape Coast in red and Navrongo in blue. And you see that the 3D7 epitope forms about 70% of the Cape Coast parasites or CSP circulating, but only about 7% in the parasites circulating in Navrongo. And not only that, these mutations that we find within the CSP epitope, some of them are found within the TH2 regions of the parasite, the C-terminal end of the parasite that was used for the vaccine development. And this will have a huge implication for the vaccine rollout in the country. So putting everything together, I'll say that interventions, whether chemotherapy, vector control, vaccine, or diagnostic, has the potential or ultimately will select for parasites that are resistant to these interventions as a result of genetic diversity that the malaria parasite develops. And these adaptations will result in drug-resistant adaptations like delayed clearance, multi-gene or cryptic resistance, vector adaptations like insecticide resistance, behavioral shifts immune and vaccine adaptations like antigenic variation, immune evasion, reduced vaccine effectiveness, and transmission resilience like increased gametocyte production, persistence in partially immune hosts, and sustained transmission. So these short-term gains, I'm saying, will have long-term evolutionary costs to the malaria elimination strategy. So what we are doing is winning battles by treating infections, but the drug pressure select for parasites that adapt. And if we do not have that mindset of thinking about adaptation of the parasite, ultimately you're going to lose the war against elimination. Again, we reduce transmission and malaria burden, but the paradox is that ultimately this is intensifying selection on parasites that remain, and sporogony will continue, and we end up having adapted parasites. Again, are we going to lose the war against elimination? So what are the implications for policy and the control programs? Our control programs or our malaria elimination program, at least I know for Ghana, we are doing a lot. We are doing all that WHO is requiring, but I think that we need more integrated surveillance systems that are interoperable. We need to provide near real-time reporting, standardized data formats, data governance and data sharing frameworks, and standardized analytics that will be useful to the program. We also need adaptive policy designs. So these policies must be very dynamic, and we should be evolution aware in our decision making. And it makes sense to support research into new drugs, new vector control measures, etc. So my final takeaway message is that interventions create evolutionary pressure on the parasite population, and the parasite is adapting silently. And at the end game, what we need for the parasite is more genomic surveillance, not just for the parasite, but also for the vaccine antigens that we are not talking about. And for the antimalarials, we also need surveillance for the partner drugs. The focus seems to be on artemisinin. So if we put surveillance together with the adaptive policy, we will be making a lot of strides for elimination. So the question no longer is whether the parasite will adapt. It is whether our strategies will adapt faster. On that, I want to say thank you, and I'd like to acknowledge all the malaria team at Noguchi and partners who have worked with us over the years. Thank you. Over.
All right. So thank you so very much, Anita. And thank you to Joelna and Anna. I think I've been a very bad host. Unfortunately, it is 4 o'clock and our time is up, so I could not allow questions time. But the good news is that our speakers are all available to answer any questions that we have. So in case we have any questions, we can, we can contact Prof. Anita Gansa or Dr. Joel Naukoli or Prof. Anna Farnett, and they would definitely be ready to answer our questions for us. So with not too much more to say, I would like to say that abstract submission for ICOPA 2026 is still open up until the 27th of May. So late breaking abstract submission deadline is the 27th of May. So for those who have not submitted abstracts, please do so, and all the best. And then I would like to say a very big thank you to everybody for joining the webinar. And I hope that we've all learned something. So today we had a very good panel. We spoke about the vector, we spoke about host immunity, and we also spoke about the parasites and how all these interplay and help with their elimination agenda. So what do we need? How do we make use of those three different aspects to aid in our elimination agenda? I hope that you have had a good time, and
Linda,
I'm very sorry that
Linda, sorry, 2 seconds for a couple of announcements. First of all, I would like to thank you for hosting and having this engaging and amazing session. This is the first session we actually hit more than 100 participants. So thank you very much for hosting it. And there have been all amazing speakers, and I'm very impressed with the work that is happening on malaria. The other thing I want to announce is that we have a continuation of the road to ICOPA 2026. The next seminar will take place on the 21st of May, and it will be by Professor Kostas Mitkoglou, and it will be on the 21st of May at 2 o'clock UK time. I will send another email to remind everyone. And lastly, I think Momar wants to say something as well, but one more last thing. The recording will be available on the YouTube channel of the World Federation of Parasitologists. So you can follow us, go there, and we will put the information also on the LinkedIn. Momar, you wanted to say something?
Yeah, just to say, you already say the best thing you have to say. Thank you so much, Tassos. What I would like to say to people is I was in the Eskimate also. I mentioned it for people attending the Munich meetings. Anyway, Tassos, by the way, this is one of the guy who gave a fantastic talk and mentioned your name. So he'll be joining the ICOPA. So for people from WFP, and I expanded also, if any of those people need kind of free registration, I know it's a lot of money, I'm happy to do that. You might receive a BCC email. I send it to Kenneth, and I provide those lists. I don't know who's getting what, so I'm just going to make it blinded, but I put there at least 30 people receiving a free registration. I know some people are asking for accommodation, but for now, the money we have was really for LMIC, but not for PI. I just want to make it clear. We're trying to bring student trainees, helping them to come. But some PI might have some difficulty. But for now, for those PI, actually happy to provide them the free registration. WFP don't have a good mechanism of collecting money. We don't have a deep pocket. But I think we're trying to explore to see how we can help also other people having difficulty, but in the executive board of the WFP. So I want to say, but I'm going to stop there because there is a lot of talk here. Very excited to hear these wonderful people talk about this malaria. Thank you so much, Tata, for giving the time to talk.
Linda, it's yours, the microphone.
Okay, so back to Linda. Well, I guess this is it. So we can all look up Prof. Anita Ganser and Dr. Joel Nakoli. They are at the Noguchi Memorial Institute for Medical Research at the University of Ghana. So their contact information will be available there. And Prof. Anna Farnett is at the Karolinska Institute, and we can also look up her information over there. And please do get in touch with the speakers. They are actually really unhappy that I could not get them time for questions and answers. I think next time I'll have probably just 2 speakers so that there'll be more time for questions and answers. So thank you very much for your attention. Thank you so very much to our speakers for gracing the occasion. And thank you all very much for your audience. And do have a good day. And please do get in touch with the speakers. Thank you very much and have a good afternoon, evening, morning, wherever you are. Thank you very much.
Thank you.